Welcome. I am Meg Smith, CEO of CureAlzheimer's Fund. Thank you for joining us today to hear about how we are advancing the fight against Alzheimer's disease. CureAlzheimer's Fund has one mission: to fund research with the highest probability of preventing, slowing, or reversing Alzheimer's disease. Since 2004, we have deployed more than $210 million toward extraordinary research to end the terrible burden of this disease. Active researchers from a wide range of scientific specialties who are at the forefront of discovery guide our funding while also pursuing their own breakthrough projects with our support. At CureAlz, we focus only on making bold bets to generate knowledge that will help the entire Alzheimer's community. We do not pursue profits or financial returns. We can provide this support to the most innovative ideas across biomedical science, prioritizing new, neglected, and unfunded questions fundamental to understanding what causes and contributes to Alzheimer's, only because of the generosity of our donors. Our board of directors, trustees, and certain other supporters cover all of our operating costs. This generosity and vision mean that 100% of the gifts from our other wonderful donors go directly to our research program. We are grateful for the community of generous donors who make these scientific advances possible, and I am glad to share some of them with you today. After watching, I hope you feel as I do: incredible resolve to find a cure for everyone touched by this terrible disease.
Thank you all for being here, and thank you all for supporting Cure Alzheimer's Fund. I can tell you that it's never been a more exciting time in the field of Alzheimer's research than right now, in terms of how much we understand about this disease and our prospects for treating it. And what I'm going to tell you up front is that the way we're going to cure Alzheimer's disease, using the term cure from Cure Alzheimer's Fund, is that we're going to end Alzheimer's disease by early detection, early intervention. We have to do for Alzheimer's what we already do for most age-related diseases. We detect biological evidence for the disease, whether it be glucose for diabetes or cholesterol for heart disease. Well, now we have those same types of biological markers for Alzheimer's that we can look at with a blood test. We have blood tests that can tell us if Alzheimer's pathology is beginning in our brains. And we can see that pathology beginning. We can make what's called a biological diagnosis of Alzheimer's before there's any clinical symptoms right now with a blood test. And that's when we need to start treating. If we start treating this disease a decade or two or three in advance, the way we control our cholesterol to stave off heart disease or keep track of glucose and A1C to stave off diabetes, we can basically put a big dent in the prevalence of this disease. And as I'll tell you, the problem is we diagnose this disease too late. Well, that's going to change. And I hope by the end of this talk, you're going to hear a lot of good news, a lot of optimism. And you need to know that without any exaggeration whatsoever, the main progress we've made in this disease today worldwide has been largely due to not just the NIH and the funding there, but because of the high impact research Cure Alzheimer's Fund does. There's no bigger bang for the buck than what Cure Alzheimer's Fund funds, because they only fund the top projects in the world that have the highest impact. And I don't say that lightly, because I think the research itself, which I'll summarize as we go through, focusing on more on my stuff. But at the end, I'm going to show you a slide of all of the amazing paradigm shifts and incredible first-time discoveries that changed the field Cure Alzheimer's Fund made. I want to show those at the end because you're going to understand them better at the end after I tell you all about what we know about Alzheimer's disease and give you this update. So first, as I'm obligated to do, these are my disclosures for companies, et cetera, that I consult with. And Harvard makes me show everything, even working with the New England Patriots. So I can't, everything has to be on here. So what is Alzheimer's disease? Well, it's the most common type of dementia in the elderly. Dementia in general is decline in mental ability that disrupts daily functioning. The most common form of dementia, Alzheimer's. 7 million patients in the US, 1 in 9 over 65 in this country have this disease. But here's what I was just telling you. Alzheimer's brain pathology begins up to 3 decades before cognitive symptoms. Thus, it's possible to now give a biological diagnosis of Alzheimer's disease, meaning we're going to see pathology brewing prior to any cognitive problems. And this is when you first want to treat. If you ask how many people in this country would have a biological diagnosis of Alzheimer's, even though they're cognitively healthy, it's probably 50 million. So this says that we can find out who these folks are. And if we have safe, affordable drugs, we can democratize the end of Alzheimer's disease through early detection, early intervention. And by the way, when Cure Alzheimer's Fund first started, and I consider myself a founding scientist of Cure Alzheimer's Fund going back 20 years, that was our mantra: early prediction, early detection, early intervention. Among risk factors, age, of course, is number one, family history, having a stroke, brain injury, and sex. Women make up 2/3 of Alzheimer patients. Women are more vulnerable to this disease. It's the fastest growing segment of our population is 85 plus and over 85 years old. If you take the earliest signs of AD in terms of clinical symptoms of cognitive impairment, it's 40% to have this disease. With a current lifespan of 80 years, the number of cases is going to triple by 2050, at which point the price tag for the country will be approaching $2 trillion. This will crush our healthcare system. So here's Alzheimer at his lab bench with his colleagues. I guess back then to do science, you had to have a big mustache. I'm glad that's not the case now. Here's August Dieter, his patient, who was 57 years old with early onset dementia. And he was the one who first described the pathology of Alzheimer's in her brain when she died. Amyloid plaques inside the neurons, these neurofibrillary tangles, and lots of neuroinflammation in the brain. He didn't call them these things back then. He had different names. He called the plaques miliary bodies, but he described the pathology. And he suggested this pathology had something to do with why this woman died with dementia. And his talk did not go over well. It was 1906 in Bavaria, and it was really tough for people to believe that a mental disorder could be caused by physical changes. So we've come a long way since then. And if you look at the prevalence of the main diseases that affect humans, from breast cancer, prostate cancer, heart disease, stroke, HIV, they're all either stable or down. But Alzheimer's disease, over 20 years between 2000 and 2019, up 145%. And this trend has remained. And the elephant in the room is here, and I mentioned this earlier. We don't diagnose diseases like Alzheimer's or Parkinson's disease, neurodegenerative diseases, until the brain has already deteriorated to the point of dysfunction. In other words, if you're showing clinical symptoms of Alzheimer's in terms of memory, if you're showing clinical symptoms of Parkinson's disease in terms of a movement, you've already had massive damage to the brain that has caused this. If we diagnosed heart disease the way we diagnose Alzheimer's, we wouldn't say you have heart disease until you're close to needing a bypass. So this has to stop. We have to be able to do early detection for disease. For diseases from the neck up, right? We have to be able to do what we do from the neck down and save these organs like the heart, the pancreas, and now the brain before there's too much damage. And you have to really play catch up to try to help the patient, which is what we do now. And here's what we know about those pathologies that Alzheimer first described. Here's the amyloid plaque, this sticky, gooey material that accumulates outside of nerve cells. Most of us start to make it as age-related brain pathology after 40 years old or 50 years old. And this amyloid triggers tangles. Now, these form inside the nerve cells, and they're these wispy pieces of something called tau protein, that aggregates into these twisted filaments that choke the nerve cell from inside. And when they form and the nerve cell dies, the tangle gets spit out, and it can go to a healthy neuron and create more tangles. So I like to say amyloid's the match. The tangles are like brush fires that spread because once you make them, they propagate. Other matches besides amyloid can be headbangs. If you play professional football and had too many concussions, too many headbangs, you can get tangles by the headbangs. Alzheimer's, you get tangles because of amyloid. So Alzheimer's is the most common form of dementia, although most dementias involve tangles. It's how you get the tangles. In most cases, in humans, it's amyloid that causes the tangles, not headbangs or brain trauma. Now, these spreading brush fires keep spreading, and nerve cells start dying, amyloid's accumulating, and this causes the brain to react with neuroinflammation. If you only have plaques and tangles, this is the preclinical stage. the pre-symptomatic stage. This is the biological phase of Alzheimer's, where you don't have clinical symptoms yet. Nerve cells are dying, but once you get neuroinflammation, where the brain's immune system, consisting of cells I'll talk about later, called glial cells. These glial cells get activated, and now you go from brush fires to a wildfire, forest fire. This causes 10 times more cell death, 10 times more damage of the neural network than the two pathologies that initiated the disease. So if you've heard about the amyloid controversy, here's what you need to know. Yes, amyloid causes this disease. There's no doubt. But it does so 10 to 30 years before you have the wildfire. And by the time you have you only get the symptoms when there is the wildfire, and we don't diagnose the disease until you get the symptoms. And then we expect to just blow out the match, the amyloid, and make somebody better. Well, yeah, if you catch it early enough, as I'll show you, hitting the amyloid can be helpful, but we have to do this pre-symptomatically to end this disease in the future. And this is just one-time cell death, 10x cell death here. This is when you get the symptoms, when the neuroinflammation hits. We see brains once in a while of people who die in their 80s with no cognitive problems, and you see plaques and tangles there galore. And you say, how could this person not have Alzheimer's? And the answer is always the same. Somehow they escaped neuroinflammation. If you only have the plaques and tangles without the neuroinflammation, you don't get enough cell death to get the disease. So at this point now, you understand more about Alzheimer's disease than most people in the world, and believe it or not, even more than some scientists who study this disease who may not be keeping up with the latest brain imaging data. Okay, so what we need to do is early prediction, family history, genetics, early detection, blood biomarkers that I mentioned, digital technologies, brain imaging. But most important, once you know that you have a problem, early intervention. Stop that pathology before the onset of symptoms. For early prediction, the Cure Alzheimer's Fund has funded the Alzheimer's Genome Project, which I've directed for 20 years, and we found over 100 different genes. Back when I was a student at Harvard for my PhD, I discovered the first Alzheimer's gene, which I named the amyloid precursor protein, APP. I maintained back then that amyloid causes this disease. It's been a wild roller coaster ride, but now we can say that was correct. Amyloid does trigger this disease. The presenilin genes, which we found in 1995, the enzymes that cut the amyloid precursor to make the amyloid. So all of those three original genes that cause the early onset familial form, mutations in these genes guarantee the disease by 60 years old. They all just increase the production of the amyloid in the brain. ApoE is the most common risk factor for Alzheimer's disease, and the ApoE4 variant increases risk by fourfold with one copy from one parent. If both parents give you an ApoE4 variant, it's a 14-fold increase. But they're not fully penetrant, meaning, yes, they increase the risk, but unlike these mutations which guarantee the disease, they increase risk. Then we have a whole slew of other genetic risk factors, 90 of them that we found, that increase risk. The good news is, and this is going to be important for the end of my talk, is that only 1 to 2% of Alzheimer's disease involves these early onset genes with mutations that guarantee the disease. 98 to 99% involve susceptibility genes where lifestyle matters. And 98 to 99% of genetic susceptibility for this disease, lifestyle can help you offset your risk. Only a 1 to 2% is that basically, unfortunately, not possible. So early detection. Well, there's brain imaging. We can image the brain for amyloid and for tangles. And now we have blood tests that can tell us whether you have amyloid, whether you have tangles, whether neurons are dying, whether there's neuroinflammation. So this is early detection. In fact, here are all the different biomarkers we can look at to just assess your brain, just looking at your blood, or in some cases, your spinal fluid, to tell us what's going on in your brain. It'll be very important at the end of my talk when I tell you how we're going to do our clinical trials, because we can use these biomarkers so we know who's at risk for the disease, and we can start to intervene early and track our progress with stopping pathology in the brain by looking at blood biomarkers that tell us about that pathology. So I'll go from fire now to water. And I like to use this metaphor for how Alzheimer's disease can be stopped. I told you amyloid, we now know, is what first causes the disease. The amyloid beta protein is the little protein or peptide that aggregates and sticks together in the brain to form the amyloid. So if you think about the brain like a sink overflowing with water, but this is your brain overflowing with amyloid. Before you have symptoms, it's going to get the damage going. This water is flowing out of the sink. There are two ways to deal with an overflowing sink. You can turn off the tap. You can unclog the drain. And as it turns out, for the early onset disease, it's more about the tap. For the regular common late onset disease that involves ApoE, it's about unclogging the drain. So to turn down production of A beta, our lab has really been a leader, and it's thanks to Cure Alzheimer's Fund, who believed in us 20 years ago in supporting this project that I carried out with Steve Wagner, where we created the first drugs that we think will safely turn off the tap of A beta. And this is important because this is a statement now that we can agree on based on all the recent data. The faster beta amyloid accumulates in your brain, the sooner you get AD. So someday when we have a drug that turns off the tap, that could be like the way you control production of cholesterol to offset heart disease. You might take a statin like Lipitor. This would be the statin of Alzheimer's. And we can also think about how to unclog the drain. Now for the statin, turning off the tap, the drug we invented is called the gamma secretase modulator, okay? Those second Alzheimer's genes we found in 1995, I showed you, the presenilins, they make an enzyme that chews up the APP to make the A beta, okay? So if we can safely block that enzyme, which is shown here, and we can do that with this drug, we can turn down the amyloid beta production, turn off the tap. And not only can we use that for just turning down amyloid production in people where they, let's say, have a blood test, and you find out, oh, I'm already making amyloid. I may be 10, 20, 30 years away from symptoms, but I'm making amyloid already. You say, how can I turn off the amyloid production safely? That's what this drug's meant to do. But more, as importantly, and maybe more importantly, for that 1 or 2% of cases where they have a death sentence, these early onset familial cases where the mutations guarantee the disease, this drug will, in most cases, reverse what those mutations do and take away that death sentence. It will remove that guarantee of Alzheimer's disease. And I was really inspired to come up with this drug with my colleague Steve Wagner after meeting with a woman who was 30 years old, and she found out she had a mutation that caused early onset familial disease. Her mother died by 45 with that same mutation. She was 30. She had 2 kids who were 5 and 6 years old. And it was tough for her to deal with this. Of course, you can imagine how difficult it is to know you're going to go the same route as your mom, who died at 45 with early onset Alzheimer's. And she looked at me and she said, so you're telling me that by the time my 2 children are setting off to college, I'm not going to know their names. And it hit me so hard. And I discussed it with my colleague, Steve Wagner, and we really went at it. This was over 20 years ago. Said, let's find a drug that turns off the amyloid in her brain, even though she has those mutations. And now we have that drug. And that drug, the gamma secretase modulator, with $40 million of support from NIH and Cure Alzheimer's Fund, after 22 years of research, is going into a phase 1 clinical trial that's planned for early 2025. And if it's safe, we're going to push to get it approved for preventing Alzheimer's disease early on. So that's turning off the tap. How about clearing A beta from the brain? Can we do that? Well, it turns out you may have heard of these 2 new drugs that were approved for Alzheimer's, Leqembi, Kisunla, from Eisai Biogen and from Lilly. That's what they do. The amyloid's in the brain. It's detected by PET scan or imaging, and now these drugs basically are called immunotherapies. They're antibodies that detect amyloid. They go into the brain, and they promote the clearance of the amyloid from the brain. So in July 2023, Leqembi was approved. July 2024, Kisunla was approved. Both were approved only for the earliest stage patients because the trials were done in patients who just started to enter into the clinical symptoms after they developed the amyloid in tangles and had just enough inflammation to show clinical symptoms, earliest stage disease, not early onset, earliest stage. And these therapies were shown to but really effectively clear the amyloid from the brain within about 6 months or so. And although it wasn't a home run, there was enough significance. In other words, these patients who were just turning the corner into clinical symptoms, that the cognitive decline was slowed by 25 to 30%. And that was much better than what we already had. And so the FDA approved these drugs. Now, slowing cognitive decline by 25 to 30%, will that result in, a spouse seeing their loved one getting cognitively better? No. But what it's doing is it's blowing out all those matches, right? It's getting rid of all the amyloid that's going to cause more disease. So over time, it should slow down the disease progress, even if the effect on cognition isn't spectacular, okay? What would be better, what would be best, going back to what I said earlier, is if we could find people with a blood test, which exists, who are starting to make amyloid in their brain, they're cognitively fine, but let's get their amyloid out. And that says 50,000. That's a typo. It should say 50 million Americans. 50 million would be in this category. And in a perfect world, we'd all have the blood test and 50 million people would find out to have amyloid and they would take these two drugs and get rid of it. But the problem is it's not going to happen. These drugs are too expensive, over 50,000 per year, in some cases up to 100,000 per year. And these drugs have safety issues where they can cause brain swelling or hemorrhage, microhemorrhage, depending on the drug, anywhere from 25 to 40% of patients. So they're not applicable for use as a prophylactic in people who find out they're amyloid positive. We need safer, more affordable alternatives for this process. And this is where CureAlzheimer's Fund comes in again, because back in 2010, CureAlzheimer's Fund believed in me and my colleague, Do-Yeon Kim, when we had the idea that we could create Alzheimer's in a dish, that we could create mini human brain organoids, tiny brain organoids, that would recreate Alzheimer's pathology exactly in the same order of events that it happens in patients. And what we were able to show is that what takes anywhere from 1 to 3 decades in patients to go from amyloid to tangles to inflammation, we can make that happen in 6 weeks in a dish. 6 weeks. So that made, I remember this New York Times article said this should make drug screening, drug discovery 10 times faster, 10 times cheaper. Well, it turns out they were wrong. It made it 100 times faster, 100 times cheaper. So now, since this invention of Alzheimer's in a dish, we've been able to screen all of the FDA-approved drugs, over 3,500 natural products, asking what's already out there that's safe, that we had no idea we could use to stop amyloid or tangles or neuroinflammation. So this was another program that was funded by CureAlzheimer's Fund called 3DDS, 3D for 3D Alzheimer's model, the Alzheimer's in a dish, DS for drug screening, 3DDS. And CureAlzheimer's Fund provided about $5 million to do all that screening. We had a team of people, and now we have those drugs. So let me tell you about that. So here's this Alzheimer's in a dish, as we call it, a 3D culture model of AD using human stem cell-derived neurons, also glia, where in just 4 weeks, we see the plaques, classic plaques in a dish, and you wait another 2 weeks and those amyloid plaques cause the tangles to form. You see classic tangles, just like in the brain, but in a dish. And like I said, this has made drug discovery 100 times faster. Think about what we had to do before. One drug at a time in transgenic mice, and it took 1 year to 2 years before you knew if you had a result. Instead, we take a plate, plastic plate the size of an index card, 96 wells or 96 holes in it. Each one has a mini Alzheimer's brain organoid, and just test 96 drugs at a time. Just go in there and say, which one stopped the amyloid? Which one stopped the tangles? Which ones stopped the neuroinflammation? And catalog them. And we've done that. We've screened all the FDA-approved drugs, 3,500 natural products, and we asked now which ones can be repurposed for use in Alzheimer's disease. And the beautiful thing about repurposing drugs is that from discovery to getting into patients is 1/3 the time of inventing new drugs. So when new drugs are invented in pharma companies, they have to go through tons of safety trials, and you have to make sure they're not going to cause any harm, and many of them fail because of safety. But when you're talking about drugs that are already approved or natural products, and you're making combinations of these to hit the disease, the time from discovery to patient can just be as little as 8 years, 5 to 7 years. We showed this was possible with a company I worked with called Amylyx. They came up with a drug for ALS of 2 combined drugs that went from initial conception of the idea in 2014 to an approved drug by 2022. So this is the way forward. Now I'm going to show you how we're doing this. Okay, here's the Alzheimer's in a dish. Here's the plates I talked about with the little wells. And this is what we do. You need to understand this cell. This is you would think the most important cell in the brain for Alzheimer's is the neuron or nerve cell. No, that's the victim. Okay. The problem is, or the killer is what's called a microglial cell. And this would be a great movie because microglial cells actually mainly in the brain act as nurturers and housekeepers. Normally, this is the microglial cell here. They have these ramifications, these little appendages, and they help get the synapses pruned, the connections between the nerve cells. They help clean debris and clear it up while you're sleeping at night. But they're not just nurturers, they're sentinels. As they're eating stuff, they're cleaning up debris in your brain, they're housekeeping. They're keeping track of what they eat because they are also the protectors. They're the warriors of the brain. And if they eat a microbe like a bacterium or a virus, right, they now get a signal to say, there might be an infection. The brain's an enclosed space. We can't let this infection spread. Wipe out this part of the brain. Wipe it out. That's what neuroinflammation is. It's the brain being attacked by its own glial cells with good intentions. They sensed that there were pathogens, and they said, we don't want this infection to spread. The brain doesn't have an advanced immune system like the rest of the body with T cells and B cells and antibodies, et cetera. We got to wipe it out. And that's a good thing that it does that. But here's the problem. The other way they get triggered from being housekeepers to now becoming killers is if they eat dead neurons. Well, what did I tell you before? As the plaques are forming with age and the amyloid plaques are causing the tangles, there's cell death. Those neurons with tangles die and they spit out the tangle and the tangle kills another neuron. It's not enough cell death to give you the disease, but it is enough cell death that when these glial cells, these microglial cells eat the dead cells, they again assume it's an infection. They're programmed that way in evolution, whether they eat a bacteria or virus or any type of microbe, or they eat a dead cell. They assume that cell is dead because of an infection. They don't get the memo that we live till 90 years old, and there's this thing called Alzheimer's and plaques and tangles. They assume infection, and again, they're activated. They cause neuroinflammation, and now even though the plaques that were here that were getting cleaned caused 1x cell death, now you get 10x cell death. Now you're going to get symptoms. So think about it. The same cell that can clear amyloid for you, and that's how those 2 new drugs work, the 2 new Alzheimer's drugs that were approved. In the end of the day, they induce the microglial cells to eat more amyloid. They're like Pac-Man, and they say, eat more amyloid, right? Those same cells that we're using to eat the amyloid, if they get the wrong signals, can kill neurons instead. So it's very simple what we want to do. We want to reprogram them. We want to take drugs and natural products from our screening that can turn the microglial cells that are killing the warriors and turn them back into housekeepers. 2 for 1. Now, the new approved drugs, Leqembi, Kisunla, just get the microglial cells to eat more amyloid. We want to do both. Remove the warriors and get more housekeeping, okay? And we have what we need. We've been screening for 7 years now. We found 136 approved drugs or natural products that, like the 2 approved drugs that just got approved for Alzheimer's, like Leqembi and Kisunla, induce the microglial cells to eat amyloid and clear the A beta. We found another 105 drugs and natural products that chill out the microglia. When the microglial cells are causing cell death and inflammation, It says, no, don't kill, go back to being a housekeeper. So we like to think that what we're going to do is repurpose known drugs and natural products to reprogram these microglial cells to not only blow out the match, clear more amyloid, but put out the wildfire, stop the neuroinflammation. So we're making combinations of drugs in these two buckets. And I should mention, we also found 81 drugs and natural products to stop the tangles. We'll get to those later. We only have so much time and funding. But right now, we're focusing on drugs from this bucket to get the microglial cells to clear more amyloid, and drugs from this bucket to get the microglial cells to cause less neuroinflammation, and we're combining them. And we're going to take these combinations and we're going to test them in clinical trials where we used the biomarkers in the blood for the pathology to ask, can we see in a patient what we saw on the dish? Okay, so let's say you take one drug here that clears the amyloid, a drug here that stops the inflammation, and you put them together, and then you look in patients and you look at these both blood-based and spinal fluid-based biomarkers that tell you, that measure the pathology in the brain. And we ask in a very simple go, no go proof of concept trial. Each trial would cost about $2.5 million to do a combination and say, are we doing it? Do we have a combination of a known safe drug or natural product with another one that actually gets more amyloid cleared from the brain, blow out the match, and at the same time stops the killing, the neuroinflammation? And right now, we have, at this time, about 25 combinations ready to go for clinical trials at the McCann Center, and we're getting those trials up and going. We raised enough funds, thanks to Henry McCann, who funded our center, to do the first trial with the first combination. I think if we can do 20, 25 trials like this, we will land on a combination of safe drugs or natural products that we can democratize the end of Alzheimer's. People get the blood test. Insurance companies now cover it because it's actionable. And you say, here, take this safe drug or this safe natural product combination and nip it in the bud. So I'm seeing the light at the end of the tunnel. I'm seeing that maybe in 5 to 10 years, 5 to 7 years, that we can go from one of these combinations that reprogram the microglial cells to clear the amyloid, to stop the inflammation, and stop preventing Alzheimer's disease. And by the way, if we can stop neuroinflammation, maybe we can help patients who already have this disease too. The neuroinflammation, the wildfire, putting that fire out. So now that I explained to you how Alzheimer's works and how I think we're going to end it, let me just show you some of the major, and I mean really major discoveries CureAlzheimer's Fund is responsible for. It was the first consortium to determine how amyloid causes cell death and causes the disease. I carried out the first entire human genome-wide screen in the Alzheimer's Genome Project to find new AD genes. And we discovered the first gene in 2008 that implicated the microglial cells. No one was paying much attention to these microglia until we pointed attention to them at CureAlzheimer's Fund when we found the first gene, CD33, that said microglial cells are involved in this disease. And in fact, Nature magazine, Nature's a major journal, just did a milestone timeline piece on the 12 major milestones of Alzheimer's discovery, starting with Alzheimer himself. And I was very happy to see of those 12 milestones, my lab was in 3 of them: the discovery of the amyloid gene, discovery of the presenilin genes, and they included CureAlzheimer's Fund's Alzheimer's Genome Project discovery of CD33, first implicating microglia in 2008. We have the first and most promising drug to lower the production of amyloid with the GSM. CureAlzheimer's Fund made this possible. We had the first study to find the rare mutations in these Alzheimer's genes in 2010. We were the first to show that amyloid beta protein isn't just junk in the brain. It actually serves as an antimicrobial peptide. I didn't get to cover that today. We created the first Alzheimer's in a dish model, the first one to prove the amyloid hypothesis. It was the first time we could show that amyloid causes tangles, something we couldn't see in mice. We were the first ones to show that the gut microbiome, the health of your bacteria in your gut, directly impacts the clearance of amyloid and inflammation in the brain, meaning the gut microbiome becomes a way to control pathology. In fact, some of you may have saw that Gary Ruvkin just won the Nobel Prize from Mass General. Gary and I are co-founders of a company called Marvel Biome that Henry McCants actually put me in touch with the CEO, and we are now using gut microbiome bacteria as drugs, and we're going to put those in Alzheimer clinical trials soon. We were the first to show that the existence of tau oligomers preceding tangles and how tangles form. So these are all paradigm shifts in the field that have changed the field, that were made possible specifically by Cure Alzheimer's Fund funding, by funding high risk but high reward projects. And we also was the first demonstration of peripheral immune T cells entering the brain and causing more inflammation. That got the Morby Prize, the first Morby Prize for the best publication, David Holzman. So now let me just end quickly with what can we do now. And, I just told you while we're waiting for these meds and everything, what can we do now given what we've learned about Alzheimer's disease to reduce our risk? So one of the things we're doing at the McCann Center is we created the Brain Care Score. This is a questions, 21 questions that have to do with everything from your blood pressure and cholesterol, BMI, glucose, your diet, drinking, smoking, aerobic activity, sleep, your lifestyle. And these show the main categories here. That gives you an idea of how well you're taking care of your brain. So it's called a Brain Care Score, where this is being launched now globally. Jonathan Rosant, who's one of the co-founders of the McCann Center, is running this Brain Care Score. But the way I have summarized brain health or brain care is through an acronym I created called SHIELD. And I got so many, SHIELD has basically taken on a life of its own out there, and it's gone viral. So much so that I'm now writing a new book, specifically on SHIELD. And just in the process now of finding a publisher to work with to put this book out. So let me go through SHIELD. S is for sleep. When you sleep, this is when your brain gets those microglial cells and the other glial cells like Pac-Man to eat the amyloid and clear it away. So you want to have as much sleep as possible where you have what I call a rinse cycle. You go from deep sleep to dreaming sleep, REM sleep, at which point these little glial cells get activated and clear the plaque and debris in the brain. Handling stress. Chronic stress causes chronic inflammation. So we talk about how to deal with stress, how to be more mindful, maybe a meditation practice. I'm going to cover these aspects in detail in the new book I write. Interaction with others. Loneliness, meaning being alone and being stressed about it, increases risk for Alzheimer's by about twofold consistently across studies. So interaction with others, stimulating your brain by interacting with others, as long as they don't stress you out, of course, is very important. Exercise. You want to aim at 120 to 150 minutes per week, where you don't have to go crazy, even a brisk walk. I use my recumbent bike. Basically, when you exercise, what we showed is that the blood flow increases, right? And when the blood flow increases, it delivers a hormone from the muscle called irisin, goes into the brain, and it causes the release of an enzyme that dissolves the amyloid. And in fact, there's going to be trials based around this. Exercise also triggers the birth of new nerve cells in the brain, something called neurogenesis. This happens specifically in the part of the brain that's most vulnerable to Alzheimer's, the hippocampus. And what we showed is that if we induce neurogenesis, as well as growth factors to keep these new baby neurons alive, even if there's plaques, tangles, neuroinflammation, when you induce neurogenesis and successfully keep those new neurons alive, the mice get better. So think about that. If patients are already suffering, have inflammation and plaque, and they've already gone down that path, but by inducing the birth of new neurons, then we can actually still help them. So this is a big project now that we're doing in our lab that we're going to be engaging with Cure Alzheimer's Fund for more support. Learning new things, okay? So when you learn new things, you make synapses. There are 100 billion neurons, trillions of synapses, connections, creating a neural network. Every experience you have, every choice you make, every experience you have is recorded in your neural network. It defines who you are. Alzheimer's disease goes through and destroys those synapses. The neural network's like a tapestry that's being unwound by Alzheimer's. What you have to do is strengthen that tapestry. The more synapses you make, the more you can lose before you get into trouble. So by learning new things, whether it's just reading a book about history, a documentary, learning a new instrument, anytime your brain's involved with learning, making new synapses, you're protecting your brain. So I like to make this joke all the time. Some of you already might have heard it. If you're learning new things right now, I'm helping your brain. But please note that if I'm putting you to sleep, I'm still helping your brain. I just love that joke. Finally, diet. I mentioned the gut microbiome. These bacteria in your gut. You've heard the saying, "What's good for your heart is good for your brain." As true, or maybe truer, what's good for your gut is good for your brain. The bacteria in your gut like fiber, plant-based fiber, Mediterranean diet, they like crunchy things that are not potato chips and candy. Okay, so fruits, vegetables, whole grains, seeds, nuts, anything that's called a prebiotic or fiber. And in fact, based on Shield, we recently published with Dean Ornish, who you may know. Dean Ornish is famous for his heart healthy diet. And together with Stephen Arnold, we just published a paper a few months ago where we actually used Shield as a treatment paradigm in a trial for Alzheimer's. Now, it was a tiny trial. There was only 50 patients, 51 patients. Half of them got this lifestyle intervention based on Shield. I'll show you in a moment. Half of them did not. And this was the protocol. The diet was whole foods, minimally processed, plant-based vegan diet. We actually had the vegan meals and snacks made and delivered to the homes where the patient and their spouse could have them. So this is fruits, vegetables, whole grains, legumes, seeds, nuts, especially low in harmful fats, sweeteners, and refined carbohydrates. No ultra-processed foods, whole foods, stressing the gut microbiome health. Sleep, interaction, exercise, learning. So there was supervised exercise. They got lectures on healthy brain lifestyle for learning new things. They were encouraged to try to sleep 7 to 8 hours per night. Of course, that's not easy, but we tried to emphasize that. And then for meditation, I invented these glasses that use light frequency. I invented these about 20 years ago, and they work really well where they flash light together with music. And the light is actually enjoyable. It's multicolored. It's kind of like a light show, like a Pink Floyd concert. And there's music that goes with it. And so right now, your frequency of your brain as you listen to me and you're learning something is around 17 to 20 hertz. And when you meditate and you get really down and relaxed, you enter what's called a theta frequency of 7.83. And this is tough to do. It's tough to meditate down to this successfully. So these glasses do it for you. So we sent them all these glasses and they loved them. They used these. And at the end of the day, this is what happened. We used the same cognitive tests that we used for those 2 new Alzheimer's drugs that were approved. And what we saw was, just like those 2 new Alzheimer's drugs, we slowed cognitive decline in the people on this protocol. So if you're going up, you're getting worse. Okay, lower is good. Here's the people who weren't on the protocol getting worse over 20 weeks. This is the slope of the people who are on the clinical trial. Much slowed cognitive decline. And then using this other test, the CDR Global, this is amazing. Here's the people who are not on the lifestyle intervention getting worse over 20 weeks. This group, by this test, got better. They actually improved. Now, to me, this is like too good to be true. 20 weeks, 50 people to get results like this. We also saw by blood tests for the biomarkers of pathology that they were indicative with a highly significant p-value of removal of amyloid from the brain. We also saw improved gut microbiome. So it's a small trial, so I can't get too overboard. But boy, for a small trial getting these results, we're inspired now to do more trials, digging into different parts of Shield to see if we can do more while we're waiting for these new drugs to come online. So that's Shield. And that's a first proof of concept lifestyle intervention trial. We hope to grow on that and do more. And I just want to end with Shield shows you what to do. I created new Shield 2.0. What not to do, the don'ts. S, smoking, no. Hypertension, no. Manage your blood pressure. Infection can trigger Alzheimer's pathology. If you have recurring herpes, cold sores, you can take antivirals for that. There's a connection between herpes viruses and other viruses and triggering Alzheimer's pathology. Get all your major vaccines because they've been shown to be protective against Alzheimer's disease. Avoid excessive alcohol. Moderate alcohol intake is okay. It's shown to be protective. Avoid excessive alcohol. Manage your cholesterol levels. L is for lipids. Make sure you take care of your LDL. And finally, D is dental hygiene. Okay, I didn't show this, but in a new study we're about to publish, we showed that in Alzheimer's brains, post-mortem brain after autopsy, if you look at the difference between a brain from an older person who didn't have Alzheimer's to one who did, we see an abundance of gum bacteria in the brain. These are bacteria that are associated with periodontal disease or gum disease. So routine flossing, okay? Keep your teeth clean. Those bacteria can get into your brain just like the viruses and trigger Alzheimer's pathology. So that's SHIELD 2.0, the don'ts. So I'm going to end there. I'm going to thank Cure Alzheimer's Fund, thank all these fine people in our unit who do the great work.
I hope you enjoyed this presentation of some of the cutting-edge research that Cure Alzheimer's Fund supports. I invite you to find a wealth of additional information on our website, curealz.org. Thank you for spending this time with us to hear about the science and progress that will bring an end to Alzheimer's. Please be in touch should you have any questions about the science we fund or how you can be part of the solution.